Imagine a world in which mosquitoes, which would usually transmit potentially lethal diseases like malaria, are completely unable to do so. This is just one example of the plethora of potential applications for one of the most revolutionary technologies in modern biology, CRISPR-Cas9. To understand CRISPR-Cas9, one must first understand the basic principles of genetic modification. An organism's genetic information is encoded in a special molecule known as deoxyribonucleic acid, abbreviated as DNA. Each of the rungs in this molecule of DNA is referred to as a nucleotide base pair. There are 4 types of nucleotides: cytosine, guanine, adenine, and thymine. In DNA, A always pairs with T and G always pairs with C, and it's this sequence of nucleotide base pairs in an entire molecule that allows it to convey genetic information. Well then, what if we could change the sequence of nucleotide base pairs and therefore change the traits of the organism? This is the exact function performed by CRISPR-Cas9, which comprises 3 primary components: the enzyme Cas9, which is comprised of protein, a single-stranded guide RNA or sgRNA molecule, and in some cases a DNA repair template. In this system, the Cas9 enzyme is somewhat like a vehicle that tries to find that specific part of the DNA to be modified, while the sgRNA is the molecule that binds to that part of the DNA. Let's say, for instance, we want to modify a DNA sequence that contains the bases GTC, ATC, and G. Since we know that A always pairs with T and G always pairs with C, we can create an sgRNA molecule with the bases CAG, TA, G, and C. The sgRNA will now bind to that specific sequence of DNA. Afterwards, scientists can insert a combination of this sgRNA molecule and the Cas9 enzyme into the nucleus of a cell, where it'll scan DNA molecules until it finds a segment that matches and binds to the sgRNA. The Cas9 enzyme then cuts the DNA and forms a double-stranded break. Through the process of homology-directed repair, we can now introduce the DNA template that was mentioned earlier. The cell will then follow this template DNA, which seems to be identical to the broken DNA, but which also contains an extra segment of DNA that is actually the gene we want to insert. The cell will now insert this novel gene to repair the DNA and therefore yield our final modified DNA.